Hi everyone, in this short video, we're going to explore ToxMSRC, an advanced deep learning tool for predicting peptide toxicity. Peptide-based drugs are emerging as a significant class in modern therapeutic development, but predicting whether a peptide is safe is very challenging. Experimental toxicity testing is time-consuming and costly, yet remains essential. But in certain cases, computational models can assist in identifying potentially unsafe candidates. ToxMSRC combines several AI techniques: first word to vector embeddings to represent peptide sequences as numerical vectors; second, synthetic minority oversampling technique to handle data imbalance; and multi-scale convolutional neural network to extract features using different kernel sizes, capturing short motifs, medium-range patterns, and longer contextual dependencies in the peptide sequence. This seems to reduce information loss and ensures that both local signals and global properties relevant to toxicity are preserved. Then, a bidirectional long short-term memory network performs additional feature extraction. The features are combined through a residual connection, which not only integrates information from both, but also helps prevent overfitting and could improve model generalization. Finally, a classification layer predicts whether the peptide is likely toxic or safe. To train the model, the authors carefully curated data from several sources along with non-toxic peptides extracted from UniProt. They removed duplicates, filtered out long sequences, and ensured only high-quality ladles were used. The final benchmark dataset contained about 1,800 toxic peptides and 4,500 non-toxic peptides for training. To evaluate performance, they used two independent test sets. Test 1 had around 320 toxic and 800 non-toxic peptides, similar in distribution to the training data. Test 2 was more challenging, with 46 toxic and 536 non-toxic peptides coming from different sources and with shorter sequences. The model achieved 92% balanced accuracy on Test 1 and about 87% on Test 2. When developing therapeutic peptides, toxicity can arise from many different mechanisms. Immunogenicity and hypersensitivity are a major concern. Some peptides can trigger unwanted immune responses, leading to antibody formation or even severe allergic reactions. Proteolytic degradation adds another layer of complexity. As peptides are broken down in the body, their fragments may have new biological activities, sometimes even toxic ones. Then there are, in some cases, off-target effects. A peptide designed to bind one receptor can sometimes accidentally interact with others, causing unexpected physiological responses. Some peptides, especially those with amphipathic properties, may cause hemolysis, breaking red blood cells, or show general cytotoxicity by disrupting cell membranes. There's also organ-specific toxicity. Depending on their targets and distribution, peptides can affect the heart, liver, kidneys, or even the brain. We also need to watch out for aggregation and precipitation and check what could happen at injection sites. One can observe local inflammation, tissue damage, and this can also reduce the peptide's overall bioavailability. In some of our own previous studies with a modified peptide tested in phase 1 clinical trials, we observed that injection site pain was a real concern for patients. From my perspective, one important takeaway is that better predictions really depend on better datasets. Right now, there's no easy solution because creating high-quality toxicity datasets for peptides is incredibly challenging. Most of the peptide toxicity prediction engines available today, including the most advanced AI models, rely on very similar open datasets. These datasets focus mainly on known toxic peptides, like toxins. That means that no matter how sophisticated the model is, it cannot reliably predict key therapeutic toxicity endpoints simply because the data isn't there yet. To build truly powerful predictors, We'll need richer datasets like failed therapeutic peptides with documented toxicity, synthetic peptides with known pharmacological profiles, and clinical trial outcomes where toxicity was observed. So while current AI tools are useful as first-pass screening tools, there's still a lot of work ahead. Now, just follow the installation instructions, review the requirements, and run some predictions on your peptides. To summarize, ToxMSRC is a powerful AI tool to quickly screen peptides. While it's highly accurate, it focuses on some aspects of peptide toxicity, as the training data do not allow for more, so further testing is always needed. I really hope this tool and the concepts we discussed today will be useful for your own research and projects. Thanks for watching, and until next time, take care.